Good morning, Asima. Good morning, Mahadet. How are you doing today?
We are doing fine, thank you.
How are you doing, Kanani?
I'm doing fine too, thank you. As we all know, digestion starts in our mouth, right?
Yes.
But what exactly happens in our mouth?
Well, we all know that food is broken into very small pieces by the teeth.
Yes, that is right. But what else happens in our mouth?
We have also learned that starch is converted to simpler sugars by the enzymes in our mouth.
Brilliant. But where exactly are these enzymes found in our mouth?
And our saliva, of course.
So how can we prove or demonstrate that there are enzymes in our saliva?
I think we have to do an experiment to see that. We can mix some starch with saliva and see if the starch is converted into simpler sugars.
But there is one important question here. How can we know if the starch is converted to simpler sugars?
Well, I think I have an answer to that. We simply perform the iodine test. If the starch is converted to simpler sugars, the solution will have a light blue or brown color. But if some starch is left, the solution will have a blue-black color when we add iodine to it.
There is also another way in which we can check if starch is converted into simpler sugars. We can do the Benedict's test. If starch is converted into simpler sugars, the color will be yellow to orange.
So now, let's do an experiment to see if our hypothesis is correct.
For these experiments, we need 15 milliliter of saliva, which we've collected from our mouth earlier. And we also need 1% starch solution to see if it's converted to simple sugars by the amylase.
We also need water as a control. We need 10 milliliters of water.
We also need iodine and Benedict's solution. This is iodine. And this is Benedict's solution.
For this experiment, we need 8 test tubes. In the first 2 test tubes, we put 6 milliliters of distilled water. Arsema, will you please do that for me?
Sure.
Add 6 milliliters of water.
And in the next next 2 test tubes, we add 6 milliliters of starch solution.
And now we put 6 milliliters of saliva to each next test tubes. Karen, please do that for me.
Now we prepare 2 test tubes in which the actual experiment is done. In each 2 test tubes, we add 5 milliliters of starch solution, and again we add 1 milliliter of saliva. Now we take the seed and shake it.
Shake it sideways.
Now we let it sit for 30 minutes. Now we take the 2 test tubes with water and put into one of the test tubes a drop of iodine. And to the other test tube, we add a few drops of Benedict solution. Now, we take the test tube with added Benedict solution and we put it into boiling water for some minutes.
Now we take the test tubes with starch solution and add a drop of iodine to one test tube and a few drops of Benedict solution to the other. And we place the test tube in which we added Benedict solution and boiling water for some minutes.
Now we take the test tubes with pure starch and put a drop of iodine into the first one. and we put some drops of Benedict solution into the other. Now we take the test tube with Benedict solution and put it in a boiling water for some minutes.
Now we take the test tubes which we have done the experiment in. We put a drop of iodine to one of the test tubes. And then we put a few drops of Benedict solution to the other. Now we take the test tube with added Benedict solution and we put it into boiling water. As we can see here, the water which served as a control, when we added iodine solution to it, it had a light brown color. And when we added Benedict solution to it, and when we put it into boiling water, it had a light blue color. So we can say that the solution had not changed color.
Yeah, as we can see here, the color of the starch solution in which we added iodine turned into blue-black. And when we compare it with the control did not change any color, but the starch solution changed color. This is because the starch is not converted into simpler sugars. And the starch solution in which we added Benedict solution is light blue. When we compare it with the control, it's also light blue. It's the same.
Now, when we look at the saliva solution in which we add iodine to, it turned to light brown, just like it did in the water. There is no simple sugar in here. But when we look at the saliva solution in which we add Benedict solution, it is light blue, just like the control.
And when we look at the starch plus saliva solution in which we added iodine, it's light brown. And when we compare it with the solution in which we added iodine, which is starch, they have different colors. This is blue-black and this is light brown. The starch must have converted to simple sugars in this one because there is no blue-black color but only light brown color. Now when we look at the starch plus saliva solution in which we added Benedict solution, and when we compare it with the starch solution, this has a yellowish color while this has a light blue color. This means the starch has converted to simple sugars in this one, while this is starch only and it did not convert to simple sugars.
So, what can we conclude from this?
We can say that our saliva broke the starch to simpler sugars. There is an enzyme called amylase in our saliva. Thus, the amylase converted the starch to smaller and simpler sugars.
So we can say that our experiments supported our hypothesis. So this means that we can accept our hypothesis.
Yes.